Microbiology and cell biology. Introduction to the module. The principles of cell biology directly deal with the cells that exist in all living organisms, including humans, animals, and plants. In comparison with this, microbiology is the exploration of microorganisms that are encountered in nature, which includes viruses and bacteria. This module looks at the fundamental concepts that provide the foundations of microbiology and cell biology. Topics to be covered: microbial nutrition and growth, virus, bacterial genetics, immunity, antigen and antibody, cells and tissues, energy trading within the cell, metabolism. Microbial nutrition and growth. Microorganisms, or microbes, are generally defined by the way in which they acquire essential nutrients such as carbon and electricity. Microbial cells need to find specific amounts of carbon to survive. This will mostly be obtained by absorbing organic molecules that are taken from other organisms. This form of nutritional attainment is something that can be compared to the human body itself. In that case, foodstuff is consumed, usually from animal or plant matter, and chemically transformed into required energy. With microorganisms, they will naturally manufacture ranges of organic compounds into cellular material. Some synthetic materials provide an alternate source of carbon, which is occasionally for economic purposes. Virus. Viruses are essentially submicroscopic parasites. They are infectious agents that replicate in living cells and live in a state that embodies both living and non-living traits. Whilst they are often described in the same way as simple organisms such as bacteria, there are marked differences between them and living cells. This includes such anomalies as: they cannot be observed by using a light microscope. They have no internal cellular structure. They contain either DNA or RNA, but not both. They are incapable of replication unless occupying an appropriate living host cell. They are incapable of metabolism. Individuals show no increase in size. A virus inside a host cell will show some of a living organism's features, such as the ability to replicate themselves. However, each type of virus has a limited host range and can only invade specific living cells. Viral structure. Research performed on the tobacco mosaic virus in 1935 showed that samples of it could be crystallized, thereby establishing some behaviorisms and qualities that were common to all viruses. This meant that they could not be considered to be living and had an entirely straightforward structure. Certain types of virus have additional protection, apart from the protein shell, nucleocapsid, that holds its genetic material. There is also a membrane of adapted host cell material. Yet, most viruses are still smaller than even the tiniest bacterial cells. The viral genome. The genetic material of a virus may be either RNA, ribonucleic acid, or DNA, deoxyribonucleic acid. Either can be single-stranded or double-stranded, and the genome itself can be circle-shaped or linear and divided into parts. Each RNA virus encodes a unique protein, including linear single-strand, circular single-strand, linear double-strand, linear segmented, circular double-stranded. Genomes can vary in size, containing as few as 4 genes or as many as over 200. These genes may code for structural and non-structural proteins, with certain enzymes needed for replication. The viral envelope. Envelopes tend to occur more animal-centric viruses and protect the virus when it travels between host cells. A lipid bilayer is formed from the original host's material nuclear or cytoplasmic membrane. The covering may appear to have spikes, which allow the virus to bind to or penetrate its host cell. They generally require a moist environment to survive, so that is why they are transmitted in bodily fluids such as blood or respiratory secretions. Bacterial genetics. The word bacteria is generally associated with negative connotations, but they are an important part of life and can be beneficial in many respects. Microscopic biological cells are found in moist conditions and organic materials. In soil, there are around 50 million bacteria in every gram. They help break down matter and protect against pathogens. Some bacteria are harmful, being capable of causing infections and death. Antibiotics are medical remedies for bacterial infections. Bacterial cells consist of protoplasmic material bound by a cytoplasmic membrane. The cell walls and membranes combine to define the structure and distinctive patterns of cell arrangements. Bacterial cells may have two basic shapes, which are different enough to allow appropriate forms of identification to take place. Spherical, coccus, or rod-shaped, bacillus. Rod-shaped bacteria which vary slightly in appearance from common-shaped, vibrio, to spiral, spirillum and spirochetes. The cytoplasm or the central part of the protoplasm, this is a semi-fluid state packed with proteins and other material. Large examples may have pores or inclusion granules to allow sustained growth and proliferation under certain conditions. Protective gelatinous layers, called a capsule, can exist outside the cell wall. Some bacteria have one or more filamentous appendages protruding from the cell wall, flagella, which are used for movement purposes, and fimbriae, used to stick to matter and organs. Pili, hair-like appendages, are also used for the transmission process of genetic information, and they are exposed to interact with the host's body's cells and material. Bacterial surface structures are the areas that are most likely to play special roles in infection processes. The bacterial cell's genetic information is mainly contained in a single, long molecule of double-stranded deoxyribonucleic acid, DNA. By condensing and looping this massive macromolecule into a supercooled state, the cell solves the problem of packaging it. The bacterial nucleoid lies within the cytoplasm. That means additional RNA material is produced, and a binding process may take place on the still attached and nascent messenger RNA. For this reason, when studying bacteria, messenger RNA, mRNA, and protein synthesis appear to be intertwined. Cytoplasmic membrane. Outside of its internal structure, bacterial protoplasm is bound by a thin, elastic cytoplasmic membrane made of phospholipids and thick proteins. These proteins are abundant and perform essential functions for the bacterial cells to grow and flourish. This includes the acts of transportation and signal transduction. Prokaryotic cell membranes, these are also densely packed with proteins, so much so that minimal space is allowed for phospholipids. The cell wall encases the protoplast and locates immediately external to the cytoplasmic membrane.